Okay, let's continue our reviewer on genetics. This is going to be part 3, I suppose. Types of asexual reproduction in animals. So there are several. Let's talk about first external fertilization. So what is external fertilization? Basically, eggs are fertilized outside of the female body. So yeah, again, these are types of asexual reproduction in animals. So this doesn't apply to female humans unless engineer engineering makes it possible. But for what point, right? Anyways, um, related to external fertilization, we have spawning. So male and female fishes release their gametes near each other in the water. So this is an example of external fertilization. How about fragmentation? So single parent breaks into parts that regenerate into whole new individuals. So I believe species that um, relate to this or exercise this kind of asexual reproduction process, they're multicellulars, um, or multicellular entities, and an example is Spirogyra. We have parthenogenesis. This is reproduction coming from an ovum without fertilization. How do we compare fragmentation with regeneration? Regeneration, this is basically the ability to regrow a lost limb or a body part. So I don't think this is a type of asexual reproduction. But usually the definition is somehow similar to fragmentation in the sense that they tend to get confused with each other. So with these definitions in mind, I hope any confusion is prevented. What is fission? So this is something exercised by unicellular entities. So fission is the splitting into parts in order to produce offspring. What is internal fertilization? Contrary to external. So this is the fertilization of an egg by sperm that occurs inside the body of a female. What's budding? So this is when a new organism grows by mitosis and cell division on the body of a parent. Okay, that's it. You know what? Let's continue. This video is gonna be too short, I suppose. So the next slides will be more on Punnett squares. I see. It's been a while since I've seen this reviewer. But yeah, let's finish until DNA. Frick. My iPad couldn't load it as fast as possible anymore. That's kind of sad. Anyways, uh, 3 years in, iPad Air Gen 4 is fighting. Okay, DNA structure and function. This is from an Edpuzzle video. I'm just not sure which one. I believe it's also available on YouTube. But I'll be discussing to you what I understood from that video through another YouTube video. Kind of funny. Let's focus first on the 4 types of bases. So, yeah, essentially we have 4 different nucleotides for DNA. First is adenine, second is thymine, third is cytosine, fourth is guanine. Okay, nucleotides or monomers, I think these are the ones. Complementary base pairing, for example, blue to green, and then black to red, green to blue, red to black. So it is known that only complementary bases can pair. So A to T, T to A, C to G, G to C. And yeah, this is an example of a complementary strand. I don't remember clearly, but three bases constitute something. I'm just not sure what that's called formally. But yeah, this is an example of a complementary strand. We have A, T, T, A, C, G, G, C, and yeah, it follows. The rest is history. DNA is made out of monomers or nucleotides, um, or made from two strands. DNA is a polymer. So in a 2D visualization, so these are the monomers, and this long line that connects these monomers is known to be the sugar phosphate backbone, which is made out of phosphate, a phosphate, a sugar, and the base. That base is any of these four. And these two strands that constitute a DNA makes a double helix. And yeah, what by the way is the point for a sugar phosphate backbone? It intends to protect the outer casing, or no. It's a protective outer casing for the DNA. DNA is kind of sensitive, so there has to be some form of protection, and that's it. So what's a genetic code? This is the sequence of bases. How about a gene? A gene is a particular sequence of DNA, and it codes for a protein. What is a codon? Okay, I found the word. I suppose it's called codon. For every three bases, they constitute a codon, or a triplet. And what is a triplet or a codon anyways? They code for a specific amino acid. And what is the overall function of DNA? It makes proteins that work specific functions. So those specific functions, they diversify into the types of proteins. So there are proteins responsible for muscle movement, some proteins responsible for other things that functions in the body. Carry out functions in the body. That's a better phrase. So proteins. Proteins are a different sequence of amino acids. So we have enzymes, hormones, structural. So I believe muscle proteins fall under this category. Some proteins play an important role in the hormonal aspect of the body, as well as enzymes. Okay. Another Edpuzzle video, we have translation and transcription. So protein synthesis can be two types. I'm not sure if these are two types, but two processes. So both occur at the cytoplasm. Okay. I get it now. So this is the protein synthesis process. So yeah, there are two steps. First is transcription. So this is basically when a single gene is taken, a single gene of a DNA is taken, and it's copied into an mRNA. mRNA basically serves as a messenger, because DNA is too big to be extracted from the nucleus for duplication. mRNA instead is a solution for that. It's small enough to exit the nucleus, the nuclear envelope, and reach the place or the site where the genetic information could be duplicated. So, yeah, I think we're talking about how cancer is formed also. I think. Because it's basically a mutation of DNA. So, yeah. Anyways. Protein synthesis. Transcription occurs among prokaryotes and eukaryotes. And it occurs, for the eukaryotes, transcription occurs inside the nucleus. But for prokaryotes, protein transcription occurs at the cytoplasm. Again, prokaryotes are more simple. They don't have membrane-bound organelles such as a mitochondria. As a result, they how do they get their food? How do they survive? They have this um, complex plasma membrane that basically exercises, sorry about that, that exercises the function of a mitochondria, which is to generate ATP for cellular respiration. Okay. For translation, uh, it occurs at the cytoplasm. That's when the mRNA arrives at the cytoplasm. I'm just not sure where that specifically is. But yeah, surely the mRNA has already exited the nucleus. And that's the reason why translation does not happen inside the nucleus. It only happens like during transcription that the mRNA is inside the nucleus and it takes place. So translation it takes this mRNA strand instructions to produce the protein. So complementary DNA base pairs. Again, we have AT, TA, GC, CG. Although there's another exception, like if I think if it's when you're given U or uracil, then there's another complementary base pair for that. I might be able to explain it later, hopefully. Found it. So if you're given A, sorry, in between DNA to mRNA, always note that yes, it's still C to G or G to C, but if you're given A, then the complementary is not T, it's supposed to be U. But if you're given T, then the complementary is A. So yeah, better note that. After transcription, the mRNA leaves for the ribosome. So this is what I was trying to mean a while ago. So after transcription, which occurs in the nucleus for eukaryotes, such as humans, the mRNA leaves for the ribosome. The ribosome is technically within the cytoplasm, but definitely not in the same place as the nucleus or the nuclear envelope. So the mRNA binds to the ribosomes. Okay, codon, we mentioned about this earlier. Code for a single amino acid. tRNA. I think this is what goes next after mRNA. So tRNA basically reads the mRNA codons and brings the correct amino acids and drops them off in order. So, yeah. What determines the shape of a protein? It's the order of amino acids. What is RNA polymerase? RNA polymerase is an enzyme. It makes RNA molecules coming from the DNA template through transcription. So, transcription, DNA polymerase is an involved enzyme. So, yeah. In order to make mRNA, this is needed. Okay. What's DNA? DNA contains thousands of genes. They're too big to bring out of the nucleus. This is what I was talking about also earlier. Ribosomes, this is the site of protein synthesis. Okay. What is the site of ribosome synthesis? Just to add to our knowledge, although I didn't write that in my reviewer, I believe it's the smooth or the rough endoplasmic reticulum. It's definitely the ER, but I'm just not sure which between smooth and rough. Basically, whatever is for the ribosomes, the other type of endoplasmic reticulum is responsible for lipid production, or yeah, basically fats. Anyways, mRNA. mRNA is much shorter compared to DNA, and that's the reason why it can be exited outside the nucleus. It is only single strand, and instead of thymine, uracil is involved. That's the reason why, if we're trying to like match complementary pairs between DNA and mRNA, if you have A, then it's U. But if you're given thymine from DNA, the mRNA would be A. So that's it. Let's talk about DNA replication. So what's the first step for DNA replication? There is a separation between two strands. The unzipping is done by an enzyme called helicase. So if you're asked a question about which enzyme basically like catalyzes or starts the entire process of DNA replication, especially if there's a key phrase called through unzipping, then that's obviously helicase. What's the result of the unzipping process? A replication fork is then formed, and separated strands provide the template for creating a new DNA strand. So there's another enzyme called primase. This makes the RNA primer. A primer is a small piece of RNA basically, and it starts the construction of DNA. So DNA polymerase, another enzyme, binds to the primer. So this is the same enzyme that I mentioned earlier, this one. So it binds to a primer and makes a new strand of DNA. The direction of the making of strands is from a 5' end to a 3' end. So the leading strand essentially is made continuously, but the lagging strand, I couldn't say but. I need a better conjunction, but basically the lagging strand runs in the opposite direction. DNA polymerase can only make this in small chunks, uh, something related to Okazaki fragments. Why make Okazaki fragments in the first place? What's the importance of these fragments? They allow, this is in order to allow DNA polymerase to replicate two daughter strands of DNA. And once the new DNA is made, exonuclease, another enzyme, removes all of the RNA primers from both strands of DNA, and consecutively, DNA ligase seals up the DNA fragments to form a continuous double strand. Okay, I think this is the hardest lesson in all of genetics for me. In my entire experience learning biology, I thought cellular respiration, the Calvin cycle, and the light reactions on regarding photosynthesis, I thought those were already difficult, but this one's a little more harder to absorb, to understand. As you can see, I can only mention to you what's written in this reviewer, but in terms of explaining, in layman's terms, how DNA is actually replicated, it's kind of hard for me. But at least I touched all of, maybe not all, but all of the necessary or the most relevant enzymes at least. Because this reviewer is also based on a video that I watched, a video that our teacher posted for us to watch back in the back in the day. I mean, technically 1.5 months ago. Okay, thanks for watching. I'll see you in the next video. Next ones will be on Punnett squares and eventually pedigree analysis.